Aloha and welcome to Botany 101. I'm Angela Nishimoto. If you remember last time we went through photosynthesis and respiration, okay? So today we're going to continue with that, okay, with plant metabolism part 3, okay? So the outline of topics we're going to cover, we're going to go through cellular respiration one more time, secondary metabolites, and assimilation and digestion, okay? And here I would like to remind you that you can always contact me if you need any help, via phone or email, or come see me in person in my office, or you may also view the lectures again, okay, in terms of podcasts at the Laulima website, okay? So let's get into the lecture. Okay, glycolysis, re-examine. So glycolysis brings about the breaking up of a 6-carbon glucose into 2 3-carbon pyruvic acids, okay? And happens in 3 steps. The first step is phosphorylation, okay? So a 6-carbon glucose receives a phosphate group, okay? And ATP starts the process, also gives up the phosphate group for phosphorylation of glucose, okay, to glucose 6-phosphate, which then another ATP is then added, okay, then which adds another phosphate group to make fructose bisphosphate, okay. The second step is sugar cleavage. The 6-carbon fructose is then split into 2 3-carbon glyceraldehyde 3-phosphates, or 3 GA3Ps. Okay, so these GA3Ps are oxidized to 2 3-carbon acids, all right, and the phosphate groups are removed from the acids, and pyruvic acid is formed, okay, which results from the oxidation of 2 fragments. Okay, so here's a picture. Okay, so here's the glucose. Okay, ATP phosphorylates it. Okay, it becomes fructose. ATP phosphorylates it again, so you see these 2 phosphate groups on fructose bisphosphate or biphosphate. Okay, then this fructose bisphosphate then splits into 2 3-carbon GA3Ps, or glyceraldehyde 3-phosphates. Okay, that undergoes several conversions. Okay, ATP is harvested, okay, for each glyceraldehyde 3-phosphate. It becomes another 3-carbon compound, another one, until finally another ATP is removed, and then you have pyruvate, okay, at the end, okay? And the phosphate groups join to 4-ADP to make 4-ATP, okay? And the hydrogens are removed as GA3P is oxidized, picked up by NAD plus to make NADH, okay, what we see right here, okay? Now we have the transition to the citric acid cycle, or to the Krebs cycle, okay? First of all, carbon dioxide is removed, okay? NADH is produced, leaving an acetyl fragment, a 2-carbon fragment, okay? This acetyl joins with coenzyme A. Now, coenzyme A is a pantothenic acid and nucleotide, okay? And pantothenic acid is a B vitamin essential to cellular respiration in both plants and animals, okay? So then this acetyl joins with coenzyme A to form acetyl coenzyme A, which then enters the Krebs cycle, okay? So here we have the giving up of carbon dioxide, formation of NADH, okay? Coenzyme A joins the acetyl group, forming acetyl coenzyme A, which then goes into the Krebs cycle, okay? Okay, now the Krebs cycle, okay? So we have acetyl coenzyme A joined to 4-carbon oxaloacetic acid to make a 6-carbon citric acid. Okay, so you have a 2-acetyl coenzyme A joining to a 4-carbon oxaloacetic acid to produce citric acid, a 6-carbon compound. A carbon dioxide is removed, producing a 5-carbon compound. Another carbon dioxide is removed, producing a 4-carbon compound. An ATP is formed, and the 4-carbon compound is eventually converted to oxaloacetic acid, okay? And some hydrogen is picked up by electron acceptors too, called NADH and FADH2. So there are 3 NADHs formed and 1 FADH2 during the Krebs cycle, okay? So each cycle uses up a 2-carbon acetyl group and releases 2 carbon dioxides, right? 1 carbon dioxide removed, a 2nd carbon dioxide removed, okay? And that rejuvenates oxaloacetic acid, the molecule we started with, okay? So by the end of the Krebs cycle, glucose has been completely dismantled, completely taken apart, okay? So here's outline of the Krebs cycle, okay? So 2-carbon acetyl coenzyme A joins with a 4-carbon oxaloacetic acid to make citric acid, which is a 6-carbon compound, which eventually loses a carbon dioxide and becomes a 5-carbon compound. Here it loses carbon dioxide, becomes a 5-carbon compound, okay? NAD plus becomes NADH, NAD plus becomes NADH, okay? It loses another carbon dioxide to become a 4-carbon compound, which becomes another 4-carbon compound, etc., until oxaloacetic acid is generated. Okay, so 3 NADH are produced, 1 FADH2 is produced. So just remember that NADH and FADH2 are just electron carriers. Okay, they carry electrons from glycolysis and also from the Krebs cycle to electron transport chain. Okay. Now, electron transport, okay? So hydrogen ions and electrons are passed along an electron transport system, okay? Some electron acceptors are cytochromes, and there are some others too, but eventually they fall to oxygen to make water. Okay, this is when water comes into the equation, okay? So during electron transport, you also have chemiosmosis happening. This is familiar to you. We talked about chemiosmosis before when we were talking about photosynthesis, but chemiosmosis was delineated in the 1960s by Peter Mitchell, a British biochemist, and he won the Nobel Prize for this in 1978. Okay, so during chemiosmosis, okay, protons are actually pumped, okay, into the intermembrane space, okay, from an area of lower concentration to an area of higher concentration, making a proton gradient. Okay, and then the hydrogen ions then diffuse back through the ATPase here. Okay, and each time that a hydrogen ion goes through this protein complex, you have the formation of one molecule of ATP from ADP, adenosine diphosphate, plus a phosphate group to make adenosine triphosphate. Okay, so hydrogen ions are pumped, or protons are pumped from the matrix of the mitochondria, the inside of the mitochondria, inside the inner membrane to the intermembrane space, the space between the inner and the outer membranes. Okay? And as electrons flow along this electron transport system in the inner membrane, the hydrogen ions diffuse back to the matrix through an ATPase, okay, forming ATP, okay? So the total, okay, for cellular respiration, aerobic cellular respiration, is 36 ATP produced, about 39% of the energy in the glucose molecule we started with at the beginning of glycolysis, remember? Remember also that during anaerobic respiration, or alcoholic fermentation and lactic acid fermentation, you only have 2 net ATP for each process. Okay, so aerobic respiration is much more efficient. Okay, will actually produce 36 ATP, which is a lot. Okay? So here we see the electrons going from one electron acceptor to another, the energy released in this electron transport system actually being used to pump protons, because remember, ions like protons and larger molecules such as glucose cannot cross the membrane unless they go through channels, okay, and they need to be actively transported, okay, whereas small molecules and non-charged molecules such as carbon dioxide and oxygen gas can freely diffuse through the lipid bilayer, okay, remember the lipid bilayer and the intermembrane proteins, okay. So the hydrogen ions actually are pumped against the concentration gradient from an area of lower concentration to an area of higher concentration into the intermembrane space, okay, and at this high hydrogen ion concentration they diffuse back through the ATP synthase or ATPase, okay, to form ATP, all right. Okay, now secondary metabolism. Okay, these are metabolic processes that are not essential for growth and development. Okay, they help the plant to survive. Okay, they result in the manufacture of colors, okay, odors or smells or aromas, whether it's nice, pleasant smells or it's noxious, toxic smells. Okay, also poisons. Okay, and other compounds that either attract or repel other organisms. Okay, so these are secondary compounds. Okay, secondary metabolites. Okay, so we humans do use secondary compounds. There have been thousands of them discovered, okay, or identified, but there may be 50,000 to 100,000 existing in plants that have, and majority, of course, not identified yet. And we use these secondary compounds for medicinal purposes, also for culinary purposes, or for medicine and for food, or to spice food, etc. Okay? So we have various categories of these secondary metabolites. We have alkaloids, okay? And one famous alkaloid, of course, is codeine, okay, from the opium poppy, and that's known as a narcotic and a pain reliever. Okay, they use lidocaine or novocaine, right, to help numb you or kill pain. Okay, that's actually also derived from the opium poppy, as well as morphine and heroin. Okay, nicotine is another alkaloid coming from the tobacco plant, of course. And tobacco, believe it or not, is also a narcotic. Okay, it's a stimulant as well. It's one of those alkaloids that kind of help you relax and stimulate you at the same time. So people who are smokers, they sometimes need a cigarette first thing in the morning to have peristalsis in their digestive tract, you know. So people who are smokers, cigarette smokers, are really addicted to it because nicotine is actually among the most addictive substances known to humans. Okay? Now, quinine is from the cinchona tree or the quinine tree, and this is actually the source of a medicine used to treat malaria. Okay, very famous. I think Jesuit priests, I believe, smuggled it out of South America. Okay, took it to Asia where they grew it in plantations, and the church made a lot of money from that. Okay, and it was also a boon to humankind because what that did is it gave us relief from malaria, a way to treat malaria. Okay, now phenolics are another group of secondary metabolites. Okay, one kind of phenolic is lignin. We talked about lignin before, right? Lignin is present in woody plants. It allows for hard wood. Okay, it allows cell secondary growth, okay, for the production of secondary tissues such as xylem tissues. Okay, so they're used to make, these woody plants are used to make furniture, of course, whether it's hardwoods or softwoods. Okay, baseball bat, construction. Okay, now salicylin might sound familiar to you. It's from the willow tree. Okay, so salicylin is actually a precursor for aspirin or salicylic acid or salicylate. Okay, and so it's a pain reliever and anti-inflammatory. Then tetrahydrocannabinol is from marijuana. Okay, and It does have medicinal uses, of course. It can be used to treat glaucoma. Okay, this tetrahydrocannabinol, which is the active principle in marijuana that gets people high. Okay, so glaucoma is it's a treatment for glaucoma. It's also a treatment for when people don't feel too good and they can't eat, you know, so it's a treatment for nausea, like when people are undergoing chemotherapy. And the third class that we're going to talk about are terpenoids. Okay. And some terpenoids of value include camphor from the camphor tree, okay, which is used for medicinal oils, okay, also used for disinfectants. Menthol from the eucalyptus tree, so this here is some menthol, okay, and here is a large gum tree or eucalyptus tree, okay, and this menthol is used in cough medicines, in Vicks Vapor Rub, in a different kind of medicinal purposes. And rubber is another terpenoid from the rubber tree, okay? And these are used to make rubber tires, of course, for our cars, and rubber bands. Okay, in this part of the lecture, the last thing we're going to talk about is assimilation and digestion, okay? So sugars, okay, may undergo many changes. Okay, some are used in cellular respiration, but others are used to form lipids or proteins or other carbohydrates, such as sucrose. Remember, that's a disaccharide, a 2-carbon sugar. Starch, which is a polysaccharide made up of many glucose units. And cellulose, also a polysaccharide, which is indigestible to us humans. Okay? So when photosynthesis is happening, the sugar can be produced in excess amounts, okay? And the plant can use the sugar to form these substances, lipids, proteins, carbohydrates, okay? And that's actually used to build protoplasm and also cell walls. So building cells is what we call assimilation, okay? Using materials to build cells is called assimilation, okay? Now, when these lipids, proteins, and carbohydrates are stored, and what they can do is they can actually be broken down, okay? So the conversion of starch to their component glucose molecules, or oils to their component glycerol and fatty acids, or proteins broken down to their component amino acids, okay? So conversion of different kind of insoluble compounds to soluble compounds is called digestion, okay? So except in so-called carnivorous plants, okay, digestion generally happens in the cells in which these proteins or starches or oils are stored, okay? So in these carnivorous plants, some digestion actually happens in the leaf part of it, as we talked about a little bit earlier, okay? Now, plants and animals use just about the same processes to do digestion, except that they do them in different places. In plants, it happens in the cells. In most animals, it happens through the digestive tract, okay? So digestion as a phenomenon is actually similar in plants and animals, except that in terms of where they actually happen, okay? So, to review what we just covered, we talked once again about cellular respiration. Okay, we talked about some secondary metabolites, okay, and we talked about assimilation or digestion, okay. And now we're going to switch gears again, okay, and get into the next chapter, okay, which is on growth, okay. So growth, outline of topics. We're going to hopefully cover growth, differentiation, and development, okay? Nutrients, vitamins, and hormones, okay? We're going to talk about plant hormones, plant movements, and hopefully, lastly, photoperiodism, okay? So what is growth? Okay? It's an irreversible increase in mass due to cell division and cell enlargement, okay? So growth may be applied to either the organism as a whole or the parts of the organism, such as the stem, such as the root, such as the fruit. Okay? So all living things start as a single cell. Okay? They increase in mass through cell division and cell enlargement. Okay? Until maybe the body of the living thing is made up of many cells. Could be thousands, could be millions, could be billions. In our case, our bodies are made up of trillions of cells, tens of trillions of cells. Okay? So then what happens? Okay? Differentiation. Okay, so this happens after cells have enlarged. Okay? It's the changes of cells to more specialized types. Okay? So cells develop different forms depending on the function that they're actually doing. Okay? So there are some cells that will become conduction cells, such as xylem cells or phloem cells. Remember vessel elements, tracheids, parenchyma cells, ray cells, fibers, right? Some cells become part of the phloem, which are the sieve tube members, the companion cells, the ray cells, the parenchyma cells, and fiber cells. Okay? Other cells may become epidermal cells. Other cells become ground tissue. Okay? If it weren't for differentiation, the body of a plant or the body of an animal would just be just a blob of undifferentiated cells. Okay? Now, development. Okay? Development is coordination of growth and differentiation of the cell into multicellular tissues and organs. Okay, so what regulates development? Okay, what determines where and when growth and differentiation occurs? Okay, it depends on the organism's genes, and it depends on the environment. Here's the nature and nurture thing again. Okay, the genes will depending on what genes are activated. Enzymes are produced, and enzymes control every biochemical reaction that's going on in the plant and in a human being as well. Okay, so the genes kind of determine what proteins or what enzymes are produced. Okay, the environment actually provides a stimulus, whether it's the internal environment or the external environment that produces the stimulus that actually leads to the production of the enzymes. Okay? So the internal environment, okay, that influences the development and growth of plants include nutrients, okay, vitamins and hormones. So nutrients, what do? They furnish elements and energy for organic molecules. And of course, plant gets these nutrients from the air and the water from the soil. Okay, so air, water, and soil. All right. Now vitamins, okay. Most vitamins are coenzymes or part of coenzymes. Okay, what they are, they're organic molecules that help enzymes with reactions, and they're synthesized in membranes and the cytoplasm. Okay, so here's looking at enzyme. Okay, here's a cofactor, here's a coenzyme. Okay, so the coenzyme helps the enzyme to do its job, and this is the site, the binding site in here. Okay, so most vitamins are produced by plants. Okay, some are produced only by animals. An example of this is vitamin A. Okay, vitamin A is actually only animals make it. Okay, it does not occur in plants, but plants actually have carotenoids, which are precursors for vitamin A. So when we eat our carrots, of course, that leads to the production of vitamin A in our bodies, and we convert carotenoids into vitamin A, okay, which are converted into retinoids, okay, and other eye proteins which help us see better. So carrots are actually good for your eyes. Okay. Now hormones are produced mostly in actively growing parts. Okay, they're organic substances produced in small amounts in one part of the plant and transported to another part of the plant where it has its effect. Okay, the effect that it can have can be like causing stems to bend, okay, or it can even inhibit growth. And this is a little image of an auxin. Okay, so we're going to go into some plant hormones, but first I'm going to talk a little bit about how they actually work. Okay, how they work is they bind to receptors, okay, in the plasma membrane, right? That causes the release of messenger molecules into the cytoplasm, and this causes a whole cascade of events to happen, okay, until a molecule is formed. Okay, so ultimately an enzyme is produced or activated, which leads to reaction in the cell, which ends up in a response to the environment by the plant. Okay, so the ones we're going to cover today are auxins, gibberellins, cytokinins, abscisic acids, and ethylene. Okay, so there are only a small number of plant hormones regulating development in plants, and each hormone can have many effects, which kind of makes it hard to tease out specific effects of each hormone. Okay, so generally you can see from these structures here that all of them are ring structures. So this is an auxin, this is a gibberellin, this is a cytokinin, this is abscisic acid, and this is ethylene. So you can see that all of them, except for ethylene, has ring structures. Okay, ethylene is a simple 2-carbon molecule. Okay, so others are all more complex than that. All right. So first of all, we'll take them in order. Okay, so auxins. In 1881, Charles Darwin and his son Francis, okay, noted that coleoptiles bend toward light. What are coleoptiles? They're sheaths, okay, that protect the shoots of the seedlings that are germinating in grass family members. So these are the coleoptiles. It's like a little sheath protecting the growing point. What they noted is that coleoptiles have a way of bending toward the light. We talked about this before, okay? And what they found out is that if they cover the tip with a little piece of metal foil, it doesn't bend, okay? And then if they remove the cap, the bending resumed. Okay, so what they did is they concluded that the coleoptile tips are sensitive to light, that they might produce something, okay, that actually makes them sensitive to light. Okay, in 1926, Fritz Wendt, okay, cut coleoptiles from oats. Remember, oats are a kind of grass, right? All right. So what he did is he then placed them on agar. Agar is a jelly. It's like a jelly or a gelatinous substance that's derived from red algae. Okay. So you place these cut oat coleoptiles cut side down on agar. Okay. Then he cut the agar up and put the agar on coleoptiles that he had cut off. Okay. If you put the piece of agar straight on the coleoptile tip, on the cut tip, if it was straight on, the coleoptile grew straight up. But if you put it off center, it bent away from the side that the agar was on. Okay. So he found out that the more coleoptiles he used, okay, the more response he got, more bending. Okay, so he and other people suspected that something was produced at the coleoptile tips, which moved into agar. So here's a little look at agar in a petri dish. It's like a little jelly. I think Japanese people use agar to make kanten, like for New Year's time and ryokan. Okay. Anyway, it moved into agar, and it was responsible for the bending away of the coleoptiles, bending away from where the little square of agar was placed. So somebody finally named it auxin, okay, because they suspected that it was linked to plant growth. Auxin means to increase. Okay, so auxins were the first plant hormones discovered. Okay. And they're produced mainly in apical meristems. Here's an apical meristem. Okay, in buds. Okay, in actively growing young parts, even part of the root. Okay, so it's kind of hard to predict exactly what the effect of applying an auxin will happen to do. Okay, because what you can do is you can actually apply a certain concentration of auxin to a plant, and it will promote shoot growth, right? But the same amount, applied to the same plant might inhibit root formation. Okay, so it depends on the concentration, it depends on the location where you apply it, depends on other external factors. Okay? So it can stimulate cell enlargement by increasing the plasticity of cell walls. It means the cell wall can expand, but it can't shrink again after it's been expanded. It's like irreversible expansion of the cell wall. Okay. It can also trigger other hormone production, especially ethylene. Okay. It's actually implicated in stem growth. Okay. It's responsible for cell division in the cambium. It's also responsible for differentiation. And it also delays fruit and leaf abscission. Okay, what is that? Abscission is when the fruit or the leaf forms a layer, an abscission layer, and eventually the plant organ falls off the plant. Okay? It also delays fruit ripening. Okay. It can inhibit lateral branching. This is something we talked about before. Remember when I had the plant and I said that you can actually cut off the tip and the side branches will come out? Okay, so auxins produced at the tip of the branch can actually inhibit the branching of lateral shoots. It can stimulate root formation. Okay, there's a rooting hormone called root tone. Okay, so there are different formulations of rooting hormones made from auxins. Okay, so they can come in powder or paste or liquid form. So you can actually get a stem, okay, dunk it in some kind of rooting hormone, and that actually encourages and accelerates the growth of adventitious roots. Remember what adventitious roots are? Adventitious roots are roots that grow where you don't expect them to grow, right? They allow for uniform flowering and fruit set. Okay, what they'll do is they'll spray fruit trees for this, and then later they'll spray the fruits, okay, to prevent premature fruit drop. So what they can do is they can pick all the fruits all at one time. Okay, they can hire people to go into the orchard and pick all the fruits all at one time, thereby saving money. Okay. It can also result in the production of seedless fruits if the auxins are applied before pollination happens. Okay. Now, auxins are also used to make herbicides, to do weed killing, okay, usually broadleaf plants, not grasses. They're not really sure why the broadleaf plants die and the grasses usually don't with auxins, but they're used as herbicides. One of them was called 2,4,5-T, which was banned in 1979. It was a component of Agent Orange. Okay, so Agent Orange is a 1-to-1 mixture of 2,4-D and 2,4,5-T. Okay, and this Agent Orange was used to defoliate the jungles of Vietnam and Cambodia, other places in Southeast Asia. Okay. Anyway, so here you can see a chemical structure. Okay, here you can see the planes kind of spraying the areas that they're going to defoliate the jungles. I think the jungles of Vietnam have still not yet recovered from it. All these, how many years later is this? Decades later. Okay. Not only that, but let's see. Leukemia, miscarriage, birth defects, liver disease, and lung disease in lab animals are linked to Agent Orange. Okay, of course they do experiments on animals with these poisons. And it's called by something called TCDD, which is a dioxin. So it will actually kill lab animals in doses of a few parts per billion. Okay, in very small doses. Very dangerous. Okay. Now we'll talk about the gibberellins. Okay. Now in 1926, Eiichi Kurosawa of Japan saw a disease of rice which he called bakanai, which means foolish seedling disease. Okay. What happens is the rice grows twice as tall as normal rice does. Yeah, but the stems are weak. So the stems collapse and the plant dies. That's why it's called foolish seedling, bakanai, right? And this is actually caused by a fungus, Gibberella fujikuroi. Okay, so that's what gibberellins are named after, the fungus that they were first noted to be reactive to. Okay, so the extracts of the fungus applied to the stem will actually produce the same effect. Okay, so gibberellins come from immature seeds, from root tips and shoot tips. Okay, fungi. Okay, so they result in faster growth in flowering plants. Okay, they don't seem to have an effect on conifers very much. Dwarf plants become normal size, like here we have a dwarf poinsettia. Here's one with stem elongation caused by gibberellins, or GA. We call it GA. Okay, it can induce flowering in flowering plants. It can break dormancy of buds and also seeds. It can allow growth at lower temperatures, such as if somebody in a temperate area sprays their lawn early in the spring, they can actually get it to green up 2 or 3 weeks earlier than normal if they spray GA on it. Okay. You can eliminate stratification. There are some seeds of like lettuce, okay, some temperate area seeds like lettuce and some grasses that need to be frozen or nearly frozen, need to be exposed to cold temperatures in order to germinate. Okay, so when we humans do it, it's called stratification. So if you apply GA to the seeds, you can eliminate stratification. You don't need to do that. Okay, it can increase the yields of sugarcane. It can result in very large seedless grapes. I don't know if you've seen the grapes in the supermarket nowadays. Whoa, they're big. They're almost as big as your eyeball or something, you know. Larger fruits, and what happens is that the fruits are spaced along the nodes, right? So what these seedless grapes do, these grape plants do, is they have longer internodes to allow for larger fruits, okay? They can also increase the length of the petioles of celery. Remember that the petioles of celery are the part of the celery that we eat. Okay, so those are about the gibberellins. So you can see that all these hormones have multiple effects, right? And they overlap each other. And they interact with each other too. Now, by the end of the 19th century, the botanists suspected that there was a substance that controlled or regulated cell division in plants. And in 1913, Gottlieb Haberlandt, okay, of Germany, discovered that some kind of phloem chemical stimulated cell division. Okay, it initiated the production of the cork cambium. Okay, Gottlieb Haberlandt. Now, in 1941, Johannes van Overbeek discovered that the coconut milk, so-called milk, or the liquid endosperm, remember endosperm is a part of the seed, so this is actually a coconut seed, and this white meat is actually solid endosperm, along with the embryo, and the liquid that would have been in it would have been liquid endosperm, okay? So you have the seed coat, you have the embryo, and you have the endosperm, okay? So you found out this coconut milk actually increased the growth rate of tissues and embryos, okay? They still didn't know what it was, though. And in 1955, this substance called kinetin was extracted from heated herring milt. That is not a misspelling. Milt is actually sperm or fish sperm, okay? So kinetin was extracted from heated herring milt, and this does not occur naturally, okay? But it actually showed that some simple chemical could promote cell division, okay? And finally, in 1964, they isolated a similar substance in corn kernels, okay? And they called it cytokinins, okay? So these are made in root tips and germinating seeds, okay? And if auxins are present, cytokinins promote cell division, Okay, so they interact with each other and they have a synergistic effect. Okay, they're also involved in the enlarging of cells, the differentiation of tissues, which we'll talk about in a little bit, development of chloroplasts, the stimulation of cotyledon growth. Here's a dicot with 2 cotyledons. Okay. And also delays senescence in leaves. Senescence means aging and dying. Okay, like SENE refers to aging or old. Okay, so a delay of senescence in leaves. Okay, now cytokinins are used in tissue culture. So cytokinins and auxins are used to induce differentiation and make whole plants from a few cells. The first people who actually did this actually used cells from carrot root. You got just cells from carrot root, you put it in a nutrient solution. Okay, they actually kept it moving, I think, and they kept it like that, and they were able to make whole plants from pieces of the root. Okay, so what goes into it is that you need to have de-differentiation of the root tissue into a mass of cells, okay, called the callus, and then re-differentiation of the callus into shoot and root. Okay, so galls actually form from cytokinins and bacteria that are produced by bacteria. So galls are actually like growths on plants that are abnormal. So galls are undifferentiated cells in a mass, and what is that also called? It's also called cancer, when you have undifferentiated cells that don't have any function. Okay? That's why people like to study these things, because it's interesting and can maybe solve some age-long problems. Now, cytokinins also prolong the life of stored vegetables. Okay, now abscisic acid. In 1949, Torsten Hemberg, a Swedish botanist, found out that a dormant bud substance blocked the effect of auxins. Okay? He called them dormins. Dorm means to sleep. Okay? In 1963, okay, 3 groups of researchers from the United States, from Great Britain, and from New Zealand discovered a growth inhibiting substance. Okay. And they named it abscisic acid. Okay. So it's synthesized in plastids from carotenoid pigments, and it's common in fleshy fruits. You know, fleshy fruits oftentimes are orangey or reddish in color to attract animals to eat them, right? So what they'll do is they'll prevent the seeds from germinating while the fruit is still on the mother plant. Okay? It'll inhibit cell growth. Okay, it also helps leaves to respond to water loss. Okay, in a drought, this abscisic acid will move from shoot to root. Okay, and increase root growth, and that increases water uptake in a drought. Okay? Now the last hormone that we're going to talk about is ethylene. Remember, that's a simple molecule, just 2 carbons and 4 hydrogens. Okay, in 1901, a Russian student named Dmitry Nebyulov, I believe that's how you say his name, he was growing pea seedlings in a dark lab that was lit by gas lamps. Okay, what he saw in these pea seedlings were decreased stem elongation, increased stem swelling, and abnormal horizontal growth. Okay, when he placed these seedlings in the fresh air, they grew normally. Okay, so he concluded, or other people concluded too, that the gas lamps in the lab produce ethylene, which causes abnormal growth in the pea seedlings. Okay? Now in 1934, R. Gagne showed that ethylene is produced by fruits. Here's a fruits from green, banana fruit from green to almost overripe. Okay, so ethylene is produced by fruits. It's produced in flowers, seeds, leaves, and roots. And there's generally a surge, okay, of ethylene that happens after tissues are damaged, okay? So if you have a disturbance in a green field, okay, what happens is that like a storm blows through, or animals, a whole bunch of animals cross the field, okay, the plants will produce fibers and parenchyma. Remember, parenchyma is a primary growth kind of cell that actually aids in support, right? And cell elongation was inhibited, so that led to the growth of shorter and sturdier plants. So this is like a response to an external stimulus. Okay, so this ethylene that was produced caused cell elongation to be inhibited, caused the plants to produce fibers and parenchyma, so you ended up with short plants. Okay, now ethylene can trigger its own production. Okay, if you introduce small amounts to tissues, a tremendous response oftentimes results. Okay, flowers fade, leaves abscise, fruits ripen, like bananas, mangoes, honeydews, other melons. Okay, and citrus fruits will actually color up. Okay. That's when you have a kind of like a greenish orange that's ripe, but what they'll do is they'll give it a whiff of ethylene and it'll actually make the orange more orange in color. Okay? Apples can be in the market all year round despite the fact that they harvest them in the fall. Okay, what they'll do is they'll put sort of sub-ripe apples in warehouses, okay, in cool temperatures with low oxygen concentration, low humidity to slow down the respiration rate. That's why we can have apples all year round, even though they're usually harvested in the fall. Okay. Ethylene can ripen pears and peaches. You know, when you get those little gift boxes with pears wrapped up in foil or in tissue paper, it's actually to help the fruit ripen. And it can induce flowering in pineapples. Okay, it can also cause members of the pumpkin family to produce more female flowers. Okay, so if you have more female flowers, you'll have more fruits, right? Okay, now tissue culture. Okay, so tissue culture is when you grow plants in bottles or jars or test tubes in the lab. Okay, with tissue culture, you need agents that allow for root and shoot development. Because generally what you're doing with tissue culture is that you're proliferating tissue. So you can actually get from plants, you can get a part of a root, as in carrot, or you can get part of a meristem, as in orchids or other plants, part of the stem, part of the leaf. Okay, you put it in a liquid medium and you put it in a shaker. What that does is that it actually disorients the tissue, so it doesn't know which way is up and which way is down, right? So what happens then is that it'll continue to grow because it's in a nutrient medium, and what it'll do is it'll proliferate, it'll make more and more cells, but these cells are undifferentiated cells. They have all the genetic information that the plant needs to make roots and shoots, but it's not doing that because it's on the shaker and it's kind of moving around, disoriented. So you can have proliferation, and people in tissue culture will take that, put it in a laminar flow hood, cut it up under sterile conditions, replate them into test tubes or into flasks, and then you can add hormones to the medium, the agar medium, and then you can get root growth or shoot growth, depending on whether you give it auxins or cytokinins. Okay, so you can have them differentiating into roots or buds and making stems. Okay, in tissue culture. Okay, because remember we talked about before that you can actually grow plants in soilless medium, or you can also do it in the lab, you know, with agar. Okay. Now we're going to talk about plant movements. Okay. So we said in an earlier lecture that plants do move, even though generally the movement is undetectable unless you use time-lapse photography, usually. Okay. So you do have growth movement in young parts of plants. Okay. This comes from internal stimuli. Okay. The kind of plant movement that was noted by Charles Darwin was called natations. Okay, and that's actually growth movements. Okay, so what he did is he tied a little sliver of glass to the tip of the plant, and he put carbon paper just above it. Okay, and as the plant was growing, he was actually making these little circular movements, making markings on the carbon. So in this way, he was actually able to show that plants made spiraling movements when they grow. Okay, that's called a notation. Okay. Now plants sometimes do nodding movements as well, such as members of the legume family, the Fabaceae. This looks like a golden shower tree. Okay. So such as golden showers, such as green beans, what they'll do is that when they're germinating, the hypocotyl, or the part under the cotyledon, kind of nods from side to side as the seedling pushes its way up through the soil. It helps the progress of the growing tip. Okay, through the soil, it helps to push the growing tip through the soil so it actually moves, and that way these nodding movements. Okay. And we also have twining movements. Okay, so gravity and contact may affect these too. Okay, so in morning glories, you have stems. There are cells in the stem differentially elongate. That means on one side, the cells elongate more than on the other side where it's shorter. Okay, that ends up in spiraling movements and spiraling growth. Okay, tendrils, twining, is also from cell elongation more on one side than on the other. Okay, that influences the differences in growth rate on one side of the stem and the other. You get visible spiraling. We also have contraction movements. Okay, remember we talked about this before? Bulbs of some flowering plants, okay, will have contractile roots, right? Like lilies. Okay, so if you plant a lily seed at ground level, you know, right near the top of the soil, by the time the plant is mature, it may be 4 to 6 inches deeper in the soil. Okay, the lily plant or the bulb. Okay, also banyan trees, their roots show contractile movements because the root, when it contacts the soil, actually contracts to pull it straight up to make the root go straight from the branch to the soil. Okay, so aerial roots of banyans do that. Okay. Now, nastic movements are movements that don't result in an organ turn towards or away from a stimulus. Okay, these are non-directional movements. All right. So you just have differential growth or turgor changes in the cells. Okay. One type is called epinasty. Okay, so epinasty often happens with a permanent downward bending. Okay, often happens in the petiole of a leaf. This is a response to an unequal flow of oxygen through the petiole or to the presence of ethylene. Okay. Now some movements from external stimuli. We have the tropisms. Okay, so this actually is the growth of a plant towards or away from a stimulus. Okay, gets greater stimulation on one side. Okay, so first of all, you have perception, where the plant part gets greater stimulation on one side of it. Okay, the second part is called transduction, where the hormone becomes unevenly distributed in the organ. And then you have asymmetrical growth because you have more cell elongation on one side and less cell elongation on the other. Okay, so you get uneven or asymmetric growth. Okay. Now, one kind of tropism is phototropism, which is you can have positive phototropism or bending toward the light, like this coleoptile tip. Okay, or you can have negative phototropism, which I guess happens in roots where they grow away from light. We also have gravitropism, which is a response to the presence of gravity. You can have positive gravitropism, where it's actually growing toward gravity, okay, like roots. And you can have negative gravitropism, like what you see here. Gravitropism and geotropism are the same things. Okay, So positively gravitropic, growing toward the gravity, and negatively gravitropic, growing away from gravity. Okay. Okay, then we have thigmotropism, which has to do with contact with a solid object. You have the coiling of tendrils or the twining of climbing stems. Okay. Chemotropism, okay, where you have chemicals like pollen tube growing into the flower, which actually sends chemicals from the ovary to where the pollen tube is on top of the stigma. Okay. You have thermotropism, where weeds will grow horizontally when it's cold and vertically when it's warm in response to temperature. Okay. Some other kind of tropisms are traumatropism, which is a response to wounding. Electrotropism, which is a response to the presence of electricity. Scototropism, which is a response to darkness. And aerotropism, which is a response to oxygen. Okay, so we actually have these aerotropisms actually happening in those in that Engelmann's experiment. Remember the Engelmann's experiment when you had the filament of Spirogyra, the prism shining divided light onto it, and the oxygen-loving bacteria hovering near the blue and toward the red area of the spectrum. Okay. Okay, so that was quite a mouthful that we just talked about, right? So this is all that we have time for today. So let me encourage you again to contact me and also to review the lectures, you know, to review the book and review the lectures and your notes. And we'll see you next time. Okay? Bye-bye.